Okay, so there's no microphone, right?
No, that's just that.
It's just not.
You want to like stand in front of it?
Yeah, I guess so. Hi everyone, and welcome to the. Oh, hi. Oh, hello, and hello on Zoom to the BML seminar series. I am not a member of the Stackowitz Lab, but I am so excited to announce Gina Chaput for this week's seminar. This is from Jay, by the way, so if it sounds weird, it's just just kidding. Gina got her PhD at the University of Massachusetts and did a postdoc at North Carolina State University before being recruited to UC Davis by Jay Stackowitz and Jonathan Eisen. She's a postdoc on a seagrass microbiome project where she's become the engine behind a collaboration involving UCD, UC Merced, and the Joint Genome Institute. She's developed seagrasses as a model system for studying marine plant microbe interactions. You may have seen Gina around BML over the past few summers, where she has been working on the role of microbes in seagrass ecology and adaptation to climate change, including the effects of these microbes on carbon storage in seagrass sediments. What distinguishes Gina is her ability to really work across fields from microbiology to genomics to field ecology. This allows her to do the sort of really novel kind of work that will push this field forward. In addition to her science, Gina is an outstanding mentor for undergraduates. In recognition, she's mentored over 25 students and several senior thesis projects. I will pause for applause. In recognition of her achievements, she was recently, like yesterday, awarded the UCD Chancellor's Award for Undergraduate Mentorship. Please welcome Gina. We're super excited that you're here, and thank you so much.
All right. Well, I'm really excited to be here. So thank you for having me. I know there's a lot on your plates in recent times with everything that's going on with the director search. So the fact that you're making time for this means a lot. As Anya said, my name is Gina. I work with Jay Stackowitz as well as Jonathan Eisen and also Maggie Sogan, who's at UC Merced. And today I'm going to be talking about unraveling the eelgrass microbiome from controlled lab experiments to field scale patterns. And across the three labs, the way I approach my research is pretty holistic, where I really try to do from the meadow to the lab and back. And so I run about, I want to say it's somewhere between 10 and 13 different projects. These all fall under three major categories. So it's the omics in the field, which I'll go over omics in a little bit. We look at plant microbe interactions in microcosm experiments. So back at UC Davis in controlled experiments to really tease apart that plant microbe interactions, and as well as microbial culturing. So establishing a culture collection of microbes associated with seagrass that then can feed back into our microcosm experiments. And then those microcosm experiments, hopefully we can then translate back into the field and see if what we're doing actually makes sense. So I'm going to first talk about omics in the field, particularly the seasonal effects project. And to do so, I just need to go over blue carbon. Now, most of you know this, but for those who don't, blue carbon is carbon dioxide that is taken in by seagrass and is stored either through biomass accumulation, both in the leaves and roots, which is then later buried into sediment as detritus, but also as exudates of dissolved organic carbon compounds. Now, my background originally is in terrestrial systems. And so when we think about terrestrial systems and this idea of plant exudates going into the soil, usually a lot of times we're thinking about more of this carbohydrates or easily accessible carbon compounds such as sucrose. So super really easy for microbes to break down, consume for carbon and energy. And in terrestrial systems, generally speaking, soil tends to be a lot more abundant in oxygen. And so that sugar can easily be taken up by microbes and processed and consumed, and then that carbon goes right back into the carbon cycle. Maggie Sogan published a paper a couple of years ago looking at this dynamic, but for seagrass, and they found something really, really interesting. So the first part is oxygen is not as abundant in sediments as, again, most of you know at BML. So under these anoxic conditions, there's also these compounds being released by the plant known as phenolics. So phenolics are compounds that are harder to break down for microbes. You can see that benzene ring right there. That is very, very stable. And so it's much harder for microbes to break that apart and consume it. And so between these aromatics and the anoxic environment, Bacteria in seagrass actually are not taking up that sucrose. They're inhibited. And so not only do we have this carbon storage or carbon sink of blue carbon, but it's actually a very sugary and sweet storage of this carbon. Now, the study was mostly done on Posidonia oceanica. I've been saying that like 3 times in the car today, different ways. So this is what happens when you're talking to yourself in the car, as well as a few other species of seagrass. And what was really interesting in the study was for the first 3 species, this was sampled in the Caribbean as well as the Mediterranean, but Zostera was sampled in the Baltic Sea. And it's only in the supplemental, and I've gotten to talk to Maggie about this quite a bit, is that it's one time point versus the other seagrasses had a seasonal aspect and a day and night aspect. Zostera was sampled once in August, and what they found that it was almost 3-fold less in sugars at the peak of summer compared to these other seagrass meadows. And I got super interested in that because my PhD was in anoxic environments looking at bacteria breaking down phenolic compounds. So I was like, oh, hey, this is awesome. I'm super curious as to whether it's because it's just a Baltic Sea and this is just an environmental situation, or if this is more Zostera specific, or if there's something else going on with the microbes. So in collaboration with graduate students, Diane and Carolina, most of you know Carolina. Diane is at UC Merced, and I believe last summer ran a tank experiment out here, so some of you know her as well. So in this project, we were looking at the seasonal aspect as well as the carbon aspect. So again, I know I'm speaking to a lot of people who know about this, but for Zostera, we know in the spring, you may have some seedlings. A lot of you see that at Tomales Bay, and eventually they grow up. And during the summer, we have this huge plume of lots of vegetative growth and flowering. A lot of the seeds we collect are sometimes between June and August. And by the end of August, the seeds are gone, and we're starting to see senescence and dormancy occur in the winter. And so I wanted to know, okay, well, knowing that there's potentially this really big sugary sink of carbon, potentially, or maybe not, and there's a seasonal aspect of the life cycle of the plant, what is really happening below ground? And so Diane was interested in that. Carolina was also interested in that. Diane was also interested in phyto hormones. So if you're interested in that kind of plant growth promoting, please reach out to her. And Carolina was very interested just in general understanding metabolomics when it comes to seagrass and this plant microbe interactions. So if we're looking at seagrass, and I mentioned before, we have carbohydrates, so sucrose, starch, cellulose, pectin, etc. And then we have those phenolic compounds. And some examples here are zostric acid, rosmarinic acid, caffeic acid, which was in the Sogan lab study, and then other polyphenols. So again, those really hard or recalcitrant carbon. And the hypothesis that I put together was during the spring and summer, you have a lot more sunlight, right? And because of that sunlight, you have a lot more photosynthetic activity, which means you're going to have a lot more oxygen released from the root tips during that newer growth. So you're going to have a bit more of an oxic environment, and you're also going to have a lot more carbohydrates being deposited from the plant, any excess, like sugars, for instance. And so that's going to be a huge boom of carbon that is utilized by the microbiome. There is still your phenolics, so they're still being produced, but they probably aren't the major source of carbon. Versus during the fall and winter, we have less sunlight. And so your photosynthetic activity decreases, the plants are going into dormancy. And so you're not going to have that big boom of carbohydrates. Instead, you're going to have a lot of detritus. And so all that plant material is being buried into the sediment. And so now you actually have a boom of phenolic compounds, especially during the fall, and some carbohydrates within that tissue as well. And so the idea was maybe during the fall and winter, phenolic compounds are what driving and sustaining the microbiome during this low carbohydrate source. So to design that, we sampled across the year, and this was back in 2022. We did, I think, a pilot in 2021 because I had never done field ecology in my life. And I was like, I would like to test going out to Mason's Marina. I have never worn waders. That was my first time doing that with Jay. So I just wanted to test it out. But when we got to 2022 and we had done the pilot study in early 2021, we kind of knew what we were doing. And so we decided to pick times that were different in sunlight and not super different in temperature. So what I'm showing you here is the 2022 daylight hours for fall, so February and sorry, fall, and then winter of November. The average daylight hours are around 10 hours. For spring and summer, it's around 14 hours. And then for temperature, yes, we do have fluctuation in Bodega Bay. However, I picked times that ideally across all 4 seasons, it's around 10 to 12 degrees Celsius. So trying to avoid having a temperature effect and really focusing more on light availability. We went out into the field, we went and collected plants, and we took the rhizosphere. So just in case I haven't said this earlier, rhizosphere is the sediment that is closely adhered to the rhizome and roots of the plant. So think about like about a 1 centimeter radius of sediment all around that area. We collected that, and just with the sediment, we did 16S rRNA amplicon sequencing, metagenomics, metatranscriptomics, and metabolomics. So all the omics, which we will get into. I'll go through basically what that entails and what kind of data we're getting from. I'm not going to go over metabolomics. That data is coming in this fall. Carolina and one of our undergraduates, Ava Andrews, is currently working on a pilot study to develop a pipeline to analyze that information. So for those not as familiar with amplicon sequencing, what this essentially is we take all the sediment from the rhizosphere, which contains a bunch of bacteria. We extract the total DNA. So that's all of the bacteria and all their DNA. And what we do is we target and copy the 16S rRNA gene. And that's essentially like the fingerprint to identify the taxonomy of bacteria. And so using the amplicon data, not only can we figure out the taxonomy, but we can also determine relative abundance of these microbial community members during that time period. And so looking at these abundances, I can plot that on a PCOA. So what I'm showing you here is each dot is a sample, and it's looking at the microbial composition. If dots are closer together, they're more similar in microbial composition. The farther away, they are less similar in microbial composition. And what's really great, at least I was very happy when things happen like they're supposed to, in my head at least, is that we see this really beautiful clustering of fall and winter overlapping a lot. The dispersion is much greater in summer, so there is a little bit of overlap there, but spring, generally speaking, is slightly shifted to the side. So we are seeing some differentiation with the summer and spring relative to the fall and winter, and we do see statistical significance with Permanova. And so the question is, okay, this is all the microbial composition. What might be driving that? What's making those changes? And so I was like, oh, we'll just plot the relative abundance and see this beautiful different patterns between the seasons. And honestly, that's not the case. So for those who are not as familiar, it's relative abundance of the microbial community. Each color is at the family level, so it is slightly more zoomed out. But generally speaking, I could probably draw a straight line across each color, suggesting that there's not a big change at the family level of these microbial communities. But the yellow is all falling under unknown. So what that means is our amplicon data is not able to determine beyond the family level. In fact, it's just getting to the phylum level, and that's as good as you're going to get. And so that's really challenging because now we really don't know, phylogenetically speaking, how different these bacteria are under this umbrella. And these might be the drivers, but we have no way of knowing that. And this is where metagenomics comes into really some handiness in that. So for metagenomics, it's the same idea. It's the total DNA. It's the same DNA that we extracted for the amplicon sequencing. However, rather than targeting one gene, we're now targeting all of the DNA and sequencing chunks of that DNA. And so we call those DNA reads or short sequence fragments. And depending on how they align together, we can group or bin that into genomes. So for this example here, the gold bars are all different reads from one organism, and they overlap in a line that we can bin into a gold metagenome. By metagenome, metagenome, MAG. So what that means is metagenome assembled genomes. We call these MAGs. So they're representative of what that isolate contains for its genomic potential. It is not fully closed, so there are some missing pieces to that, but it does give us more insight than sometimes we can get from 16S. So this tells us who's there, just like amplicon sequencing, but this also tells us the potential functions that they can do. And then the next question is, not only is there potential functions, but what are they actively doing? What makes them important possibly in these different seasons in relation to the plant? And so with that, we can do metatranscriptomics. So from the same sample of sediment, we extracted the DNA, got our 16S and metagenomes, but now also we have the total RNA. And same idea is that we're taking different chunks of the RNA. And now rather than mapping it to itself, we're going to align or map it to the MAGs. And so for this example here, I'm showing 2 gold bars that align to the same gene on the gold MAG, and that is going to give us a gene expression count of 2. So we can use these counts as an idea of the gene expression or activity of certain microbes in the sediment that are interacting with the plant during these seasons. All right, so going past the technical fun stuff of this, now we can start really looking at our hypotheses. So I'm going to be discussing the metatranscriptomic data, which again is that gene expression, that activity of the different bacteria present. And we're going to first look at this idea of the phenolic compounds and how the plant's production of these phenolic compounds are interacting and being consumed by the microbial community. So to do that, what I'm showing you here, again, in the top left corner, just a reminder, this is META-T data. What I'm showing you is a heat map. And the darker red it gets, the more of that expression of a particular gene. In this case, each column is a pathway. So, and these pathways are related to phenol compounds. And then each row is a season. And so it's a little loud, but I'm going to break it down a little bit. What caught my eye initially, and hopefully is now catching your eye too, is when looking at the spring and summer, we have these big red chunks at the tail ends of our heat map. And looking more into these pathways that have high expression or a lot of those RNA reads are related to catechol, benzoate benzoyl-CoA, and protocatechoic acid. As a microbiologist and someone who studied aromatic compounds, I was like, oh, I know what that is. And what it mostly was is that it's looking at the beta-catechoic adipate pathway. This is a very common pathway for bacteria to generate a lot of energy and food. And so these compounds produced by the plant are being immediately uptaken by bacteria. There's a lot of different ways for it to consume that. What caught my eye on the other hand, though, was something I had never heard of, which was this gentisic acid, because it's only expressed during the spring and a little bit during the summer. And I was like, what is this? And come to find out, there are papers, specifically from the 1980s, that started looking at this type of compound being produced by active growing young seagrass. And it's actually a deterrent for a lot of grazers, such as amphipods. And so we're seeing this uptick of bacteria consuming this compound when there's the most newest growth starting to occur for these young seedlings and for this vegetative growth. So that was really interesting and not what I expected to find. We're still obviously diving much more into this, but this is just a first glance at this data. I honestly just finished analyzing it like 5 days ago, so we're still kind of really diving into this. When looking at the fall, there's this beautiful big red block in the center, which is really interesting. And we're still seeing catechol, benzoate, procatechoic acid just through different pathways. So there's multiple pathways, like I said, that bacteria can use this. We're seeing some shifts of how they're using that. But what also caught my attention was these compounds, caffeic acid, cinnamic acid, and camaric acid. And this ties back way back to my PhD. And I was like, oh, wow, okay, we're doing this. And this is these are derivatives of these compounds here. I'm gonna step away for a second. Camara alcohol, feral alcohol, and snap alcohol. These are the building blocks of lignin. For those who don't know what lignin is, a complex polymer that is used part of the cell walls to protect the plant. So you might have heard of cellulose, hemicellulose, and lignin. That is the outside of this plant's protected environment and pathogens and all this other stuff. So this is a very complex matrix. And so this kind of suggests that during the fall, which we expect a lot of detritus deposit, that now we're seeing a huge uptick of potential lignin compounds being present in the soil sediment for microbes to start breaking down. Again, we're still working on this, but it is a suggestion that we are kind of seeing that trend of detritus being deposited and the microbes interacting with that. When it comes to the winter, though, kind of radio silence. There's a little bit of a expression where, for instance, the lignin derivatives are still being slightly consumed going into the fall, into the winter. But generally speaking, it's pretty dormant. And that could be because this is the closely adhering rhizosphere. So potentially detritus and other compounds are seeping further down below in the sediment, and so they're just not available to the microbes anymore, or that there's just limited exudates at this point. So just generally pretty quiet during the winter at the microbial level as well. When it comes to carbohydrates, again, we were expecting a lot of activity of carbohydrates during the spring and summer and limited activity in the fall and winter. Um, Again, we're looking at metatranscriptomic data. So the darker red there is, the more expression and more RNA mapping to certain pathways. And what was very interesting, for me at least, was that we're seeing actually not a lot of activity of these really easy, accessible sugars at spring and winter. And that instead, we had this big boom of carbon consumption of these carbohydrates during the summer and fall. And so kind of thinking about it, just kind of, again, starting to really think about this data, is we can kind of go back to the life cycle and kind of think about it, is that during the spring, yes, there is some vegetative growth, but really you're dealing with seedlings coming out of dormancy, and they're not going to be producing a lot of carbohydrates. So really that boom of carbohydrates is going to be during the summer, and then that's going to continue into the fall with the detritus of leftover carbohydrates from that material. And then by the winter, that's no longer available to the rhizosphere. And so a slightly different story, not so much light driven, but more of the life cycle. And so kind of just to recap regarding this project is that we have seasonal phenol compounds are shifting across seasons with indication that life stage might play a larger factor. So seedling versus vegetative versus senescence. And carbohydrates are really having maximal vegetative, are having a peak of consumption during maximal vegetative growth and detritus deposit. And generally speaking, whether it's phenolics or carbohydrates, winter is relatively quiet. In regards to that original idea of what we're kind of looking at here is to be determined. So a couple things you need to still do is looking at the metabolomics. So actually see if there's sugars constantly present throughout the year or if they are going down or up across seasons. Also, the idea is that we are looking at expressed genes, but there might be a whole pool of other genes not being expressed that are also related to these carbons. And so what that means is that they have a bunch of genetic potential, but they're not actually being active. And that would be very similar to the Sogan paper. In addition is also who's expressing what. So what mags are these and at what abundance of mags. So for instance, you might have a certain bacteria that's very low in abundance that has a ton of expression, or you might have a more even amount of different bacteria that all have low levels of expression. And so again, that changes the story of actually what is occurring in the microbial community. Okay, so that is the omics in the field. I'm now going to briefly go over microcosm experiments in the next 10 minutes or so. And so when thinking about microcosm experiments, I was more interested in this idea of looking at microbes as a tool for seagrass restoration. And so what I'm showing you here is a paper looking at the 10 golden rules for restoration of seagrass. And one of the things they really pointed out was future-proof your projects. So thinking about resilient plants and resilient seeds. But I also think that means a resilient microbiome. So for those who've ever had an upset stomach and took probiotics, your gut was not happy and therefore you suffered as the host. And so you also need a very resilient microbiome to function as an organism. Well, when it comes to the microbiome role for restoration, we know very little in regards to seagrass. But this is not the case in terrestrial systems. In fact, it's a huge industry when thinking about crops and drought resilience and pathogen resilience, where they have taken crops, understood the microbiome, again, looking at mags, looking at culturing, and designing these synthetic microbial communities that they inoculate on the plant so that way they're more resilient to droughts and pathogens and such. And that can be implemented at the seed level, or it can be implemented at the adult level with drones spraying certain bacteria. So for those who might be more familiar with wheat and fusarium, they tend to spray wheat after the fact that they're already blooming, grown, so that way the fusarium cannot attack the seeds themselves. But for seagrass, we do have some basic understandings. We do know that, for instance, what I'm showing here on the left is that there are distinct microbiomes. So in yellow is the sediment, in red is the roots, and then in blue and slightly silver is leaf and water of seagrass. So there are these distinct compartments that we can look at microbiome composition. We also know that microbiomes have various roles, whether it's carbon, nitrogen, or sulfur. And we also know that depending on the environment, that both the host and the microbiome are responding. So if there's a temperature, like a heat wave, for instance, type of stress, not only is the host responding to that, but the microbiome is also stressing out and responding to that heat wave. And then there's even been some more preliminary evidence, which is really exciting, just came out recently about dipping seagrass roots in plant growth promoting bacteria and planting them in the field and having a higher success of restoration. So there is already evidence of this being possibly as a tool. And I like to say, I like treating seagrass like a crop. I want to make it more economical. I want to make it more, how can we, sounds kind of like how to capitalize on this essentially, of like really thinking about how to get more industry and other people interested in preserving seagrass, not just as a keystone species in these ecosystems, but also as an ecosystem service. So this is all really important. But everything I just showed you is predominantly adults. And what I'm interested in is the seed. So there's very limited knowledge regarding the seagrass seed microbiome. I think one paper just came out recently on Zostra, and that's the first one I've seen. But again, in terrestrial systems, we have a very good understanding of this. You have your seed microbiome that came from the flowering plant. So that's your vertical transmission of microbiome, so from mother to seed. Then we also have this introduction to the soil microbiome, right? And so there's that horizontal gene, it's not horizontal gene transfer, sorry, horizontal transfer of the microbiome that is going to change the trajectory of that microbiome assembly as it establishes as a seedling all the way to the adult. And so I really want to start kind of having that fundamental understanding for seagrass. And I could do that using this device here in collaboration with LBNL, which is called the EcoFab 2.0. And so what I'm showing you here, I'm gonna just step away, is this device is autoclavable. So it's completely sterile on the inside. We can use synthetic seawater that is filter sterilized, so everything is aseptic. The top part is your water column, and then below we actually have about a 2 millimeter chamber that only has an access point of where the seed actually is, so you can have this really nice root development. I didn't want to deal with hydroponic effects. That's actually a huge issue with a lot of this type of studies. So what we did is we constructed hydrogel beads, which match the same porosity that you would see in sediment. And so that way the roots can have as much as possible natural development in a type of sediment that we can see through with microscopes, so we can actually look through that. You can also pull samples down below from these ports here if you're looking at metabolomics, which we are, but I'm not going to show that today. And so, yeah, so the question was, can we actually manipulate the seagrass microbiome in these chambers? So for the first year of my PhD postdoc, I'm tired. Through the first year and a half, I was mostly focusing on developing this with Trent Northern and Pete and Deer, just trying to make this as possible, manipulating the microbiome and also having a stable EcoFab chamber. Because again, these were originally designed for terrestrial systems. So we had to deal with positive pressure of water being inside, leakage, and all these other things. So we learned a lot. And the main experiment that we ran was this idea of having seeds that either had a reduced microbiome. So they went through a series of washes that removed a majority of the epiphytes or bacterial epiphytes that are the bacteria on the outside of the seed coat, or they were intact with synthetic seawater, and so they maintain that epiphyte microbiome community. We then did destructive harvesting across different stages. So stage 1 is initial seed germination. Stage 3 is the initial first true leaf production. And then stage 6, which has your roots and 2 true leaves, and the cotyledon has been fallen off. And we did this destructive sampling for metabolites as well as that amplicon sequencing that I talked about earlier to look at the general community composition. Okay, so again, we're saying this is reduced microbiome, so we'd like to prove that. And so what we found was that with the reduced microbiome, it is indeed reduced. It has a lower and significantly lower alpha diversity when comparing to the intact seeds. And if we look at the beta diversity, regardless of the life stage you're at. So this is including stage 1, stage 3, and stage 6. It's a completely different trajectory when you're reduced versus if you're intact. So we are seeing this long-term effects of the trajectory of this microbiome. And again, I'm interested in what's driving these differences. So we can look at differential abundance between these two conditions. And so we can do that like this, where basically I'm showing you in red the different bacteria that are more abundant significantly in the reduced condition versus intact, and the ones that are significantly less abundant in reduced microbiomes compared to intact. And so to plot that, we can now look at those relative abundances. So what I'm showing you here is a phylogenetic tree of these different red dots. And then across the way is looking at the relative abundance. So the larger the circle, the more abundant they're present. And then their color is based off of the class that they're assigned to for taxonomy. And we're going across stage 1 through 6 for intact and stage 1 through 6 in reduced. Okay, so the main trend I want to pull away from here is if you have an intact microbiome, the microbes you start with at stage 1 pretty much stay the most abundant all throughout the life stages. They don't really change. Versus if you reduce the microbiome, you start off with these guys and then they disappear and some other guys show up and then they disappear and then some new guys show up. And so it's much more transient in the microbiome establishment. And the other part I want to point out is these microbes in your reduced is whatever was still inside the seed. So really what you're working with are these endophytic bacteria. So they're just filling whatever role is necessary that usually the outside or epiphytic bacteria may be actually having a larger role, typically speaking. Okay, so yes, we showed that we can successfully manipulate the microbiome. The next question is, can we actually detect shifts across the different stages of growth? And so again, I'm looking at stage 1, stage 3, and stage 6 with these differences in plant morphology. It's the same experiment, but this time we're just going to focus on the data from the reduced seeds. So this is seeds that started off with a reduced microbiome and we went through the whole growth stage depending on where we destructively harvested. And then again, we look to see how the microbiome composition is changing over those life stages. And what I was very happy to see is we see this beautiful movement from stage 1 to stage 3 to stage 6. So there is this shift of who is playing what roles in the microbiome and how they're holding their abundances and composition over time. And to look at what's really driving that, it's the same idea. This is your your taxonomy here. And then this is the abundance plot, bubble plot. And what we really found was that as you start from stage 1 all the way to stage 6, you're increasing in gamma protobacteria and you're decreasing in bacillus. So basically your bacillus is very high at the beginning and slowly peter off. And what's really interesting especially is that you're seeing this uptick of growth promoting bacteria such as Helmintholaceae and Methylphagiaceae that are increasing as the plant is growing. And that makes sense because you're going to need plant growth promoting bacteria as you're growing. So it is matching what we were hoping to see, that at this very fine level, I mean, we're talking only a couple weeks in between these stages, we can start seeing really definitive differences at the microbiome level. And so in the context of thinking about the other types of ideas for synthetic microbial communities, we can start picking out members that may have an important role and really start thinking about how we're going to isolate those and manipulate those to see how that's going to affect long-term on the host and its microbiome trajectory. Okay, so last part in my 2 more minutes, because I really want to give them credit, is the culturing aspect. So I designed a Swabs to Genomes, a course back at UC Davis. This is a course-based undergraduate research experience. I drive out to Bodega Bay to Mesa Marina with my undergrads, and they collect seagrass tissue, come back to the lab, and then basically swab or isolate bacteria from seagrass leaves, seagrass roots, and seeds, and both the epiphyte, so surface level, or endophyte within tissue level of these different bacteria. This has been over 20 students that have been part of this. We have about 650 different isolates, and of those, there's about 72 genomes, which the students are analyzing. So I'm very big about they get to see from the start to the end. So not only are they isolating these bacteria, they get to do the DNA extractions, they get to run the gel electrophoresis, we send it off for sequencing, and then using platforms like KBase, It's an easy way for students to learn how to actually assemble genomes from RNA reads, get the phylogeny, run pangenome analyses, and start working on first author publications. So right now we have about 4 or 5 different students working on their own first author publications regarding this work. And we can use these isolates to then go back to the whole idea of microbial communities. So with a select 4 that we have that were collected by these students, we are sterilizing or reducing the microbiome of seeds as well as the roots of adult plants. And we are then inoculating them with these 4 isolates, putting them back into bodega flow-through tanks, and then seeing after a certain amount of time, as well as a heat stress, so a heat wave in this case, do we still see those isolates present, and do they give the hosts an advantage in regards to those heat stresses? Okay, so with that, I'd like to thank my advisors as well as our collaborators, as well as the graduate students, and then Emma Dean, who will be joining the Sogan lab this fall, but was also a lab technician in the Seckwitz lab. And then a huge thank you to all the undergraduates who are part of this endeavor. Like I said, we have over 20 undergraduates who are part of this. Yeah, I would never be able to pull off what I just did in the last 4 years without them. And they have been exceptional. Just a couple things. Andrew is looking at microplastics. Eric and Jackson have discovered, sequenced, and now we're working on a paper looking at an omycete pathogen that's killing off our seeds from Mason Marina. Varsha, where is she? She's somewhere in here. Varsha, oh, there she is. Varsha is looking at cryopreservation methods for Labby. Max and Taylor have looked at developing competition assays between Labby and bacteria. And then a lot of these students at the top are the original Swabs to Genome students who have been working tirelessly on K-base and learning more about genomic analysis. And with that, I will take questions.
Thank you, Gina. That was great. Serena and I are working together to get questions from the chat. We apologize. We don't have a microphone besides the one that's with this camera. I don't know. So I would like to ask you to repeat the question.
Yes, I can repeat the
in the room for sure.
Yeah.
But yeah, let's open the floor for questions.
Yeah,
I have a question about the, I guess, the second part of your talk.
sure.
Around that autoclavable chamber, grow chamber.
Yes.
And the transition of these reduced microbiome back sequence.
Yep.
And I'm a little confused only in so much as that it seems like there were families of bacteria that were popping up in the growth
Yes.
in a sterile chamber. So I'm not.
Yeah.
Where did all that come from?
Yep.
Not protected in the feed at first, like where?
Love this question. I do get this a lot. So to repeat the question for those on Zoom, the idea of when in the EcoFab, if this is all sterile, why are we getting random pop-ups of new bacteria as the plant is growing if there's no introduction of other bacteria? The answer to that is that 16S amplicon is only as good as its detection. So when you have your reduced seeds, we're only able to detect microbes at a certain threshold that are present within the tissue at that time. As the seed is growing, those abundances could get higher and other ones are going to get pushed out. So that's a little bit of a host effect where now all of a sudden those that were below detection are at much higher detection later on. So nothing is being introduced, but just basically the roles and the abundances are greatly shifting during this time.
So those other bacteria may be there.
They're just very low levels initially. Yep.
It's a quantity.
Yes. We do also check the EcoFab itself, like we plate out things and check the washes, and so it's not introduced. It's all, I like to view it as a seed is a suitcase, and you might not be able to find that sock that you're desperately looking for until the last minute, and then you can find it. That's kind of how I describe that. Yeah.
So you've obviously worked on a wide range of projects. What is, I guess, like maybe what are the top like 2 questions that you're really excited at looking at in the near future?
Oh boy. Okay. So the question is, across all my projects, what is the most exciting aspect or questions that I'm most interested in? So for my future lab, hopefully, is that I would like to really look at microbiomes as a tool. So I didn't present this here, but we have this idea of priority and priming experiments, again, in the EcoFab. So it's this idea that if we're going to manipulate the microbiome, I would like to know which points are more important. So is it that if you're there at the very beginning, so similar to what you were asking, if you're there even at low abundances, does that matter more because you were there first and you can fill those niches? Or is it that, so again, that priority effect, or is it more that you might have better functions and you're kind of jumping on from the sediment or other things to really drive it? And that really changes how you design microbiome experiments with microbial communities, because you need to know when to inoculate your plant to get the best result. So if you're trying to get better germination, maybe the seed isn't the answer. Or if you're trying to, maybe we should be injecting the flowers themselves beforehand. I have no idea. For priming, that idea is to prime your host itself. So the idea is that if in this particular experiment, we were looking at sediment microbiomes from Tomales Bay and Bodega, and depending on that source of microbes present, does the seagrass itself have a higher innate immune system compared to a different microbiome being exposed to it? And so will it have more resistance to heat stress or more resistance to pathogens like LABI, depending on what microbes they're able to recruit from their environment? So that's a priming type of situation. And again, if we're going to start thinking about treating seagrass like a crop, like I would like to, you need to understand these dynamics before you can come up with a tool that makes sense. And so those are big ones. So the EcoFab in general was like, okay, here's the basis of my lab. I'm not crazy. And yes, undergrads can use it. And now the more scientific aspect is, okay, now that we have this foundational method, I want to make sure we understand when's the best time to start looking at these interactions. Yeah. Yeah.
Question about this.
Sure.
It seems like a lot of, you use words, I guess, data, just aligning to expect the data.
Mhm.
Can you also use it for just
Yes.
if you get more resolution than you would expect?
Yeah, okay. So the question is, I had brought it up at 16S that we were losing some resolution. We had a lot of unknown members. And then with the metagenomics, I talked about more just mapping the meta T to it, but you're curious about, well, can you identify better taxonomy or can you not? This depends. Okay, so what's really nice about metagenomics is you don't have to bin them to mags. You can actually treat them as raw DNA reads, and you can find taxonomy through that. And that has, depending on who you ask, because there's a lot of debate with big data, it's very hard. But ideally, you should be able to detect more of those bacteria and have a bit more resolution than 16S. 16S is relying on one served region and looking at SNPs. And single nucleotide polymorphisms, so changes in the A's, T's, G's, and C's. And that idea of having metagenomics, you might have more robustness in your coverage of the DNA that's being present versus 16S is very much, I mean, I'm gonna go on a tangent. Every technique has its own limitations, DNA extraction, the machinery itself. But the argument is with metagenomics, potentially, that you are getting a deeper coverage of more unknown things, unlike 16S. Yeah.
So when you talk about restoration, it seems like the seedlings sometimes pick up the microbial community horizontally.
Mhm.
So
Yes.
if you're doing that restoration at a place or a site that's really heavily impacted, where your grass has maybe been absent for a long time and maybe the soil or sediments are really deep, like know if the appropriate pool of bacteria is actually there or horizontal transfer.
All right. So the question is, if the area of restoration is maybe greatly damaged because of the anthropogenic effects, or you have a seagrass been open not in a really long time, if you're gonna put seeds in there and they're uptaking bacteria from that area, how do you know if it's harmful, or how do you mitigate the harmfulness? Is that basically what we're saying? Okay. So this is why priority and the priming are so important, that foundational understanding. Because if we know that if you inoculate a set of microbes at the very beginning of a seed, and there is a strong priority effect, then it won't matter about that horizontal gene transfer, or gene transfer, keep doing that, horizontal microbial transfer, because the things that we put on it are going to establish and maintain their functional roles. So I don't have an answer yet, but I'm hoping to kind of figure that out before it. So I didn't know if you were making a comment.
No, I was gonna ask a question.
Oh, okay. But yeah, so that is one of the things. So with crops, for instance, sometimes it's more beneficial to inoculate the seed. Other times it's better to spray stuff on when they're adults. And we don't know that yet when it comes to seagrass of what's the best timing. So I'm hoping that with my future work that we can start kind of teasing apart what is the critical point of adding microbes that either protect the plant, or maybe honestly, granted, there's a lot of regulations, but you could try maybe implementing some sort of pre-inoculation in that sediment of certain bacteria. It works on terrestrial systems. We're not really sure if that's going to work with aquatic systems. And so like, there's a little bit of nuance here of like, I'm not quite sure which direction is the most effective and what will be okay for regulations in different countries. But ideally, we can start kind of teasing apart the best times. Yeah. Or, yep.
Yeah.
Oh, sorry, I missed the first little bit of talk, so you might have covered this, but are you doing any like mitigation measures for, like non-target, because you're talking about horizontal transfer?
Of microbes,
so
yeah.
Are you doing any studies around like impacts on outside environment on outside organisms, like non-target? So like impacts on what that might do to the microbiome of adjacent organisms?
Oh,
Biomes specialized.
I am not, but Nikki is. So indirectly, Nikki is a graduate student in Jay's lab. He's looking at the potential of, oh gosh, is it isopods or amphipods? Isopods. This is where I'm definitely a microbiologist. How isopods grazing on seagrass changes the dispersion of microbes across the meadows. I think that probably falls more under what you're interested in, because he was talking about looking at the gut of the isopod as well as the plant and like the dispersion. You're absolutely right. There is a chance that introducing microbes onto a plant and then other things eating it, how does that affect them? I will say though, we are using bacteria that are naturally derived from these environments. And so ideally there's just a higher abundance, but that does not necessarily mean that's going to be detrimental to the other species. But again, we would have to do lots of experiments on that. I personally have not started any of those. I know that's an interest in the STAC lab, as well as many others. But it is a great question to ask and think about, because you don't want to be reckless, right? There's a reason why FDA doesn't let you just spray crap on plants randomly, or sometimes they do, but.
Because alternatively, if there is a negative impact horizontally, the soil amendment would be a potential application, correct?
Yes, so if, again, just to zoom, the point is that if you can amend soil because there is no negative impact, you could possibly do that. Yes, again, I don't know about the dispersal of these bacteria and how long they actually maintain in the sediment. So what you're describing, yes, that can sometimes happen with like crops. They what is it called? Starts with a T. Yeah, turn over the soil and inoculate the soil as they're putting in new plants. And people do that in like the Amazon and stuff like that, to help mitigate, nitrogen accumulation and other aspects of that. Whether or not you could do that in a seagrass bed is a different story. There's a lot of pore water movement that's happening that you don't deal with terrestrial systems. So there's a lot of unknown that I'm hoping that the community will start thinking about, just like what you're saying is we have these techniques, we do them in terrestrial systems. Can you implement the same thing in aquatic systems and maintain a long term? I do know the seminar speaker that just came, this coral work, thank you. Amazing woman. She was saying how they inoculated coral with certain bacteria and saw that bacteria that's beneficial actually migrating to other coral that was not part of the study and having a positive effect. So there is some instances of dispersal and interaction that are occurring. Yep. Yeah. Oh, sorry.
No, you won't. You won't. No, you're good.
In your eco-pod studies, are you seeing any differences in like whether or not this is successfully germinated?
Okay, so In the current experiments, we only transfer them as soon as they germinate. We have not done what happens with seeds that we have no idea what's going to go. We do get dormant seeds. We do get seeds that are covered in that Oomycete pathogen that, again, like I said, Eric and Jackson are working on. And we only have 50 ecofabs. So I have to be very careful with how we plan this out. So we do an initial screening. This is another big undergraduate project where we have over 1,000 seeds in Petri dishes in sterile environments. So again, avoiding that inoculation issue of like contaminants. Everything's done in the biosafety cabinet in hydrogel beads. And they, through the Petri dish, look and study when they start seeing initial germination. And we have a little graph that says, if you see this, please tell Gina. And then Gina transfers them to the EcoFab.
Okay, I have a question.
Yes.
For the bacteria that you've been growing in the lab to then inoculate with the EcoFab, are there any similarities of those bacteria with the ones that you're seeing with your seasonal studies? And are those some of the ones that are consistent over time? And can this give you some clues about like whether or not
Yeah.
priority effects are happening
Yeah.
or priming effects? With priming effects, the other way I was thinking about it was like, are there some bacteria that need to come first in order to lead the way, if you will, for
Yeah.
other bacteria? And if any of the ones that you've been growing in the lab, if you can categorize in any of those ways.
I'm gonna try and think of how to like rephrase that.
Sorry.
Here. Okay. So the first question is, if I were to make a microbial community from the cultures we're growing and inoculate them in the EcoFabs, which we have not done yet, they're seeds, but we haven't moved into the EcoFab. The EcoFabs have always been either native or reduced microbiome.
Maybe a better first question is,
Okay.
are the what you're growing in culture?
Yeah.
Do you see some of those in your
Yes. That I can answer. Okay.
your seasonal samples, and like how do those match up?
Okay, so the question was, the cultures that we have, do we see those pop up in the seasonal data with metagenomics? I haven't directly looked quite yet. Yes, we see overlapping families. I would say in general, the Halomondaceae are always seen, especially when it comes to like spring and summer versus what we're seeing in growth periods. I can say, so again, I haven't directly compared to the seasonal, but I have done metagenomics on a Northern Hemisphere project, which is sampling the meristem of adults across the in the Mediterranean, Atlantic, Pacific, all those. And we do see a matchup of cultures that we have are highly similar to those found on the meristem of adult plants worldwide. And so I'm expecting to see similarly in the seasonal data. We just haven't quite gotten there yet, but that is a huge question I have about whether or not we are isolating the right things. I will say a subset of the culturing that we're doing, I did not show this up here, but we did a heat wave experiment last year. And we isolated bacteria from plants that were at ambient 3 degrees above and 6 degrees above. And we do see differences in what is popping up, at least clonal, the bacterial morphology. So we get pink and yellow isolates in the 6 degrees, we get more tan and white in the 3 degrees, and like very blasé stuff at ambient. So we are seeing some sort of effect. We're currently going through for DNA extraction, genome sequencing, and we'll be picking based off of functional capabilities and enough phylogenetic diversity to create a microcosm that we're going to inoculate on the adults and seed plants and redo the heat experiment starting next year. Yeah.
My question is around, it's the rhizosphere, right?
Mhm.
And generally it's soil around,
Correct.
as you described. You also did some culturing in the lab of essentially roots, leaves, and other parts of the seagrass, focusing on bacteria.
Yes.
Right? So I'm curious to know like more about the fungal microbiome.
Yay.
Maybe what's going on there, right?
Yeah. Oh, I wish I had that slide. Okay. I didn't because I figured I'm so happy to have a fungal person here. That we're really excited about fungus. So we did do ITS sequencing for many of these experiments. And we are seeing detection of different fungus. I will say for the seasonal ITS data, the problem with the fungi databases is that they're so understudied that most of the sequences remain unknown. So it's really hard, like statistically, there was no difference, but similar to what we're seeing with the 16S data is there's like this huge proportion of unknown. If you have suggestions of how to have a better robustness of trying to identify taxa, that would be fantastic. Because I agree that marine fungus plays a huge role in what's happening in these below ground. I know technically speaking, there's no like fungal network, but I did read recently about the dark fungi matter, which was done in, I think, Denmark, with this idea that it's not mycorrhizae, but like this dark aspect of some sort of communication or something. So yeah, I'm definitely open to ideas, but we are definitely sequencing that all the time to make sure we have that data. So as databases get better for reference, we can actually go back and look at that more.
Any other questions? Well, please join me in thanking Gina. This is great.
Thank you.